So good evening. Pleasure and delight to see such a wonderful turnout, and welcome. I'm the Dean of Faculty of Medical and Health Sciences, John Fraser, and it's my great pleasure to welcome you to the third of a series of 7 inaugural lectures for 2015. I'll just start by introducing, and then we'll have some introductory remarks from Professor Peter Stone. So, inaugural lectures, for those of you who are unfamiliar with academia, are in fact one of the highlights of our academic calendar. And as I said before, this year we have a record 7 new professors, and they will be presenting over the coming 4 weeks. So last week we had our first 2 inaugural lectures, and inaugural lectures are a tradition that dates back to medieval times. In those days, universities were very much just a collection of professors and nothing else. They were recruited into an elite brotherhood. They supported each other, protected each other. They lived in cloistered surroundings away from the real world, answerable only to themselves and to their esoteric interests, while expecting at the same time to be handsomely remunerated for just being smarter than everyone else. I'll leave it to you to decide whether things have changed very much since then. So the inaugural lecture is a tradition that has not changed in universities, and these are the public announcement of our new professors. Inaugural lectures are a means of showcasing our most eminent staff and a celebration of an event that represents, for many, a pinnacle in one's academic career. Elevation to the title of professor is not something that is taken lightly in academia. The process of selection is long and involved. It requires intense scrutiny from international peers who then confirm to the university that the individual has reached the level of the professoriate. And they are in fact an internationally eminent expert in their field, thus giving them the license to say anything they like about their subject in the hope that people will believe them. Inaugural lectures are also a wonderful opportunity to invite colleagues, family and friends to the university to learn about the journey that has led to academic success, the milestones, the decisions, even the heartbreaks, but most importantly those mentors who've assisted along the way. Tonight it's my great pleasure to introduce our third inaugural lecture, Professor Andrew Shelling. And I now invite Professor Peter Stone, who will provide some introductory remarks. Thank you.
It's a particular pleasure to be introducing Professor Andrew Shelling to you tonight. I've known him for nearly 20 years now, and his reputation went before him. In his abstract about the lecture we are to hear, he mentioned that he was a sporadic case of a genetic diagnosis of professor. Well, let me assure you, he is no mutation. He began his academic pursuits in what was physical education, what we might now perceive as sports medicine. And he certainly brought his love of physical activity, fitness and wellness to our department. And by 1999, he had firmly established a tradition that I could commend to the whole faculty, Dean. That was the tradition of Fat Dog, the fantastic annual triathlon of the Department of Obstetrics and Gynecology. Now, I have here a priceless souvenir from the 1999 event, which can be purchased for a suitable price to support Andrew's ongoing research. However, being a little more serious, Andrew moved from physical education to science whilst in Otago, and he obtained a PhD, being the top student in both physical education and biochemistry. After this, he was awarded a prestigious Nuffield Medical Fellowship, and he spent 3 years in Oxford at the Institute of Molecular Medicine. Now, in 1996, the Department of Obstetrics and Gynecology in this university was looking to strengthen and reinvigorate its reproductive science teaching and research program. And Andrew was the logical choice to be appointed as a lecturer. This position allowed him to establish his own laboratory and develop his interests, which included the genetics of reproductive cancers. In 1999, he became a senior lecturer. I was appointed head of department soon after that, and from 2001 to 2013, Andrew was deputy head of department. And we had a very successful partnership which enabled the department to be strong in both clinical, academic and basic science teaching and research. During that time, I was delighted to be able to support Andrew's promotion to Associate Professor in 2006. And along with Professor Leslie McCowan, who's unable to be here tonight, we also saw his promotion to Professor this year. Andrew has contributed to the advancement of reproductive science not only in the department and in the faculty, but very widely. He has been president of the New Zealand branch of the Human Genetics Society of Australasia, the deputy chair of the Advisory Committee for Assisted Reproductive Technologies, a very important position to discuss the ethics around all the in vitro fertilisation that we do. In addition to being on many editorial boards and international societies, as well as having an impressive publication record in a 33-page CV. Single spacing. Professor Shelling is currently the Associate Dean of Research in the faculty, the Associate Dean of the Performance-Based Research Funding, the important process by which our university research is recognised and in part funded. He's also the Director of the Bachelor of Medical Science Honours Program, which is so important in encouraging young trainee doctors to gain research and science experience and consider a clinical academic career. And just recently, he initiated the partnership with the Auckland District Health Board, termed the Auckland Academic Health Alliance, which just last week announced its first awards. This will build collaboratively on the strength of both this university and our neighbour, the major hospital in New Zealand. When I was born, the double helix had not been discovered. Andrew tells me that by his birth, we knew what a chromosome was, but now we are to hear how Professor Shelling has brought us to 2015 and he has sequenced the professorial genome. Professor Shelling, we look forward to hearing about that.
Thank you, Peter. I'm not sure I came along to the right talk, if you're talking about that person. Thank you very much for coming. I'm a bit overwhelmed by the number of people here, and it's a privilege that you can all come along and hear what I've got to say about myself. If you're here for the science, that might be next week's talk, because I'm going to talk more about myself and the journey that I've been on than about the research that I've done, although I'm going to add a little bit of research along the way. This is a photo I took just a few weeks ago. And I think a lot of us in the faculty really get to where we are by the amazing support that we get from our head of departments and head of schools and our deans. So Peter's been a great support to me over the years, a great team together, and more recently with Leslie McCowan, who can't be here tonight. There are many in the deanery I want to thank. I understand that I've got the most people coming along to the inaugural talk, so I've been keeping a close eye on that, being a little bit competitive. But that's until I discovered that most of the invites were from, most of the people responding to the invites was Mark Barrow. He seems to have responded more than once. We've got a great team in the deanery, so thanks to Trevor and Mark and Karen, who can't be here, in Paparangi, and predecessors Ross, who's here tonight, and also I can't, prior to my time in the role as Associate Dean Research, I had a great predecessor, Louise Nicholson, who was was big shoes to step into, but she helped me get set up and been a great mentor along the way. Thanks also to John for having the faith in me. 2.5 years ago I got an email, please go and see the Dean, and I wasn't sure what I had done. And I was informed that it was the day after he got back from a trip from Europe, and it was a half-an-hour appointment. And I knew that I was either in serious trouble or he was going to ask me to do something. And so thank you for the vote of confidence and asking me to be Associate Dean Research. and Ian Reid for all his wisdom and knowledge, and also Salome does a bit of the same thing as well. I just want to thank Jo, wherever she is. Jo's just there. I think Jo, if she was given a PhD, could run the place better than I could. So Jo's been a great support over the years. We've got a great team in the faculty. So Kira's here. Tarek can't make it tonight, but we call ourselves the Grant Busters. Who are you going to call? Because a lot of people in the faculty want to know who the best point of contact is. Also well supported by Research Office. Grateful for Tracy to be here, and we've got Lauren and Ruth who support us from Research Office, and of course Jane Harding, whose attention to detail is second to none. I'd also like to thank my mum and my family who are here. Mum's both my biggest supporter and my biggest critic. She gives me advice on how I should do things and giving talks and doing research, so I'm very grateful that she's been able to make it up here from Nielsen. She's his 90th birthday this year coming, so it's she's a tough old lady, and I've got a lot of respect for her. I've also got my little brother here. He was born 14 months after me, and I quickly realised he was probably a bit bigger and stronger and smarter than me, so the only way I could sort of get ahead of him was when he was about 3 years old and I was 4, I found an axe and chopped his fingers off. So that put me ahead of the game for a few years. But the surgeons seemed to be able to sew them on fairly well. But I think for a lot of us that I guess in academics we're grateful for the support team at home, and I'm especially grateful to my family. So my wife Gretchen, who's always been there supporting what I'm doing. Her mother Janice, who's here as well, and my 2 kids, who I'll mention briefly a little bit later on the talk, but it's always great to have support from your family. And there's another addition to the family. We've got my daughter Madeline has just got engaged to James, and so great to have him as part of the family. So what I'm going to do tonight is just give you a little bit of a background to who I am and where I came from, and then talk about my time at Otago and Oxford. I'm then going to talk a little bit about life in Auckland and a few other bits and pieces along the way. I'm going to struggle to keep to time, but I'm sure if I see people leaving, I know my time is up. So I'm from Nelson, sunny Nelson. I was the 7th of a family of 8. We lived out in the countryside, had a great outdoors life. So here's me as a little baby. So apart from overcoming being very handsome, I also had to deal, I was also very active, and by 9 months was wandering around and seemed to like to move around a lot. I was brought up in a part of Nelson called Hope, so always when people say where you're from, I always used to say I live in Hope. And I didn't get the joke for many years, so Hope School had a population of 28 students in 2 classes. It was a country school and things were fairly basic, but of course at that part of time in New Zealand we all got a really good education, which I'm grateful for. You may not be able to read this, but Andrew shows promise in outdoor activities seem to be the only common feature of most of my reports, and certainly the marks and grades didn't really match always my ability. I went to Waimea College where I learnt many useful things, but the thing that I discovered I was really good at and was a point of difference and something that I guess gave me a lot of confidence was I discovered I was good at running. And so sport became my, I guess, my predominant occupation at that time, and I guess I got a lot of pleasure out of it and also a lot of success. And I won't go into too much detail, but I broke all the school records at the time. I think they still stand 30 years later. When I was 15, I won the Wellington under-16 cross-country and made the Wellington under-18 cross-country team. For the next 15 years, I represented Wellington, Otago and Tasman at different times in cross-country and running, road running and athletics. Eventually made it to the World Mountain Racing Championships a few years later, in a place called Gap in France. And I think it was just a very simple thing. You had to start at the bottom of the mountain and make your way up to the top and down again. And that was the day. It was, I guess, the pinnacle of my sort of athletic ambitions. But I did note that I was by far the biggest and the most heaviest in the running team, which did show that my phenotype wasn't quite right for perhaps that event. The other thing I noted is that in the race, all behind me were orange singlets, which are the Dutch team. So even though I've got Dutch blood, the Dutch don't have any mountains. So again, it shows that phenotype can win over genotype. Growing up in Nielsen was a lot of fun. It's the mainland, the South Island. I lived a lot in the outdoors, fishing and wearing, I think they're Daisy Duke shorts, I think there might be. A few things happened along the way that I probably don't want to talk about too much. I climbed a few mountains, had a few adventures, but I was very fortunate to have a family that really, very old-fashioned, conservative and traditional, but they supported me really well. Dad never really understood university, probably too much. He understood how to work hard, work ethic, and was very pleased when I picked up my first paycheck, I think was... He died about 12 years ago from cancer. I assumed that by working in cancer you were immune to the disease, but it doesn't appear to be the case. So Mum and Dad supported us really well. Basic principles like honesty and hard work, and a Christian faith as well has always been something that supported me. Mum and Dad tried to overcome the fertility rate in New Zealand. So I was used to a very large family, so it was always fun to have lots of kids running around. So I went, as Peter already mentioned, I went to Otago University. I was good at sport. That's the only reason I went to university. And the degree I did was physical education. Fazetas were supposed to have 3 stripes and IQ of 50 was what they were. The first year wasn't very stellar. And I struggled for a little while, and you can see that one course was incomplete, and that was I passed the gymnastics part, but I failed the social dance. So I had to, that was one of the courses I had to repeat. Along the way, I managed to settle down a bit and won the Peter McIntosh Prize, which is the top student in physical education. It sort of took a little while to get the hang of study, but after a few years, everything turned out okay. Things went so well that I was convinced to do biochemistry. I was very interested in metabolism and citric acid cycles and things like that, and I carried on biochemistry, but couldn't get a project in the area that I was interested in. I did an honours, and the only available project was on genetics and looking at the integration of viruses. So for some of my students that are here, this is handwritten down the bottom here, and if David Jenkins was here, he'd probably faint because that's something nasty and toxic that I spilled in the lab. My honours went like a dream. Everything I did turned to gold, and I assumed that science was always like that. And these are some of the first southern blots that we ever done in New Zealand, and I know my family's here, but each of those bands represents a piece of DNA that has been made glowing by radioactivity at the time. And so people used to come and see what I was doing as part of my honours degree, and this was actually looking at some human cells. Each of those lines over here represents a virus that's inserted itself into a piece of human DNA. So it just seemed so natural that doing well in biochemistry, and that was at the time it was the top postgraduate department in New Zealand. So to get top of that class, I assumed that the road ahead for research was just glowing. I had a fantastic mentor. Merv Smith was a man that I deeply respect and was very supportive of my academic career. So everything was going along at that point reasonably okay, but the first 2 years of my PhD were a complete disaster. Nothing worked, and I was wondering where the magic had gone. And then just as things started to get better, I went into the library and opened up a journal and saw not what I was doing, but exactly what I was doing in the journal called the Proceedings of the National Academy of Sciences, USA. So this is one of our top line journals. I was absolutely devastated because after 3 years of struggle, suddenly my exact project was there in print. And I guess it was a moment of enlightenment for me. I decided what I was going to do. Either I was going to quit, I was going to change topic, or I was going to carry on and fight. And I guess that was a good kick up the pants for me, because everything in life at that point had been relatively straightforward, and I'd always seemed to be able to get what I wanted. So what we did is we decided we would fight, and we would do something a little bit different and carry on. So my PhD probably took a little bit longer than it should have. My mum and dad were a little bit horrified about how long I'd been at university. We were able to publish what we did in the end, and the publication was well recognised as something that added something considerably to the field and helped me with the next stages in life. This is not a firing squad. Something about then happened as well. So that's 29 years ago and a bit. This is me lined up here and my little brother over here. So Gretchen and I were married as students. I guess we broke the norm. People wait until they're a bit older to get married, but we decided we would get married when we were young. So Gretchen's always been a supporter right through my career and has always been there to look after me. I guess, I don't know whether this is true or not. But towards the end of my PhD, I decided that I would start looking at the next stage in my life. My head of department and my supervisor had both gone to the University of Oxford, and I'd always seen that as being the leading light for research and academia. About the time this Oxford Nuffield Medical Fellowship advertisement came around, and I wondered whether I might have a chance of that. It was designed for medical students rather than science students, and no science students really had it for a number of years. So I applied and was pleasantly surprised to get an interview. Flew up to Auckland from Dunedin and had an interview. The other person was a medical student and apparently thought she had it all covered because I was only a scientist after all. But I think she made one little mistake. She turned up in a party frock and said, "Do you mind if I go first? I've got a party to go straight after this interview." So perhaps that wasn't the best appearance. When my interview came, was probably second to a professorial lecture. It was probably the second most stressful thing of my life. And I was surrounded by all these very important people that told me how the Nuffield Fellows always went on to do amazing things in later life, and they did this and did that, and how much of an honour it was to get this award. It was a little bit ironic that about 25 years later I'm now on the other side of the table doing the same thing, so perhaps they could have been right in the end. I'm actually wearing a Nuffield tie. So these are awarded either year every 2 years to one Australian, one New Zealander and one South African. William Morris, he's Lord Nuffield, didn't have any children and donated all his money to medical research. Because Australia, South Africa and New Zealand bought Morris motor cars, Canada didn't, we get the awards. So I had 3 years in Oxford. They were great years. Went to the Institute of Molecular Medicine. The head of that at the time was David Weatherall. Fantastic place for research. Resources were unlimited, and I had the time of my life. I probably worked too hard, but when you're awarded a prestigious fellowship like that, you feel like you've got a duty to somebody, I don't know who, to actually do your best and do well. Weatherall also got me a teaching position at Christchurch College. The potential was just be a postdoc and not have any attachment to the university at all. That was a great experience. Some of you might know this hallway from Harry Potter, and it was a great place to be and teach students. Probably the most fun experience, and I could talk for a little bit about this, was somebody convinced me to play rugby. He figured I was a New Zealander, so I must know what to do with an oval-shaped ball. I hadn't played rugby for years. I'd been been more interested in running. And suddenly discovered I was in a team surrounded by much bigger chaps mainly from Harrow and Eton, and they couldn't work out what social position I was based on my accent, of course, so they just accepted me quite happily. It turned out they were quite good, and we actually won the college rugby competition in the second year that I was in the team. So this is the dinner that we had, lots of good food and wine, and then that night we went out around all the colleges singing our college rugby song to all the colleges we'd beaten, usually involving a little bit of beer along the way. The interesting thing was that the Bishop of Oxford joined us, and my last bit of the evening was doing a haka on the steps of the Ashmolean Museum with the Bishop of Oxford down below. I did do some work in Oxford. My project was a genetic analysis of ovarian cancer, and I worked very hard. 12 publications for my 3 years in Oxford. I just want a little bit of science in here. These are slides from, I guess, 25 years ago, just to highlight that the ovarian cancer that I was working on was no different to any other type of cancer. In terms of survival, if you pick up the cancer earlier, if it's localized or early stage, then most people survive from all types of cancer, and ovarian cancer is no different. If it's diagnosed late, I saw Ben Lawrence earlier, so he knows what I'm talking about. Diagnosed late, then the outcome is very poor. The difference with ovarian cancer is that most of it is diagnosed at a very late stage. And this slide is probably fairly true 25 years after I made it. Ovarian cancer is still diagnosed late because there's no early stages or signs or symptoms. Whereas breast cancer, endometrial cancer, cancer of the cervix are often picked up a lot earlier. So the reason that ovarian cancer is often called the silent killer is because most women will present late when the outcome is very poor. If we had better ways of detecting ovarian cancer, we could pick it up earlier and have better outcomes. So we did some very exciting work while we were there and published in journals like The Lancet and the British Medical Journal. But my true focus was on chromosome 6. And I just want to go through some of the things we did back in the old days, just more for my students' sake than anybody else. So this is a picture of chromosome 6. I'd sorted it in a centrifuge and then labelled it with a fluorescent dye, and then labelled it back onto all these chromosomes. But what I was most interested in is this is chromosome 6, one of our chromosomes. There was a little bit down the bottom that in over half of all ovarian tumours was lost. So a little piece of DNA somewhere down the bottom of chromosome 6 was lost. And I found many tumours where there was a little chunk missing right down the very bottom. So that we took that to mean it must be holding an important cancer gene. So what we did is got a whole lot of normal chromosomes. So here's normal chromosome 6. Here's its mate over here. And we made a little glass pipette and we cut the end of chromosome 6 and put them in a test tube and collected them all up. And I had to go to London for a week to get enough bits of chromosome 6 to be able to work with them further. So my students in the room, this is a gel with markers that you can clearly see. A gel run properly. So each of those little spots on this what's called an agarose gel is a piece of DNA from that region of chromosome 6 that I'd carefully cut off from the chromosome. So I had thousands of copies of those. And then what we did is we sequenced them on these special plates called sequencing gels. And we used to have to read them. So this is T, 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 C, T, 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 G, C, T, A. Write them down in a little book and then enter them into a computer. And so if I was lucky, I could do a few thousand nucleotides in a day. And working hard, I could get a little bit of chromosome 6 working, sequenced and analysed. Now the interesting thing is I could probably, so what I did in 3 years in Oxford, I could probably do in an afternoon now with the Human Genome Project on tap. So while it was fun work and interesting, it's technology and things have moved very quickly. Something else happened in Oxford. So although I'm a lecturer in reproductive science, we decided we weren't going to have any children just then. 9 months later, Madeline appeared and changed our life forever. As I was about to come back, thinking about what I was going to do next, I wrote an article that to be published in the New Zealand Medical Journal, and I'm not sure, you won't be able to read that. I'm just going to blow up a little piece. I was rejected, and I was very disappointed. And the little bit was that the article starts off reasonably enough, but I think that anyone with but a specialist in molecular medicine would soon be lost in the complexities of tumorigenesis. In other words, it would be over the head, but all but a handful of our readers. So this was my rejection letter from some research that a review that I wrote, and this is from again from just over 20 years ago, where I was talking about a cancer patient. The best way of getting a better clinical outcome was talking about things like early diagnosis, rational drug design, gene therapy, which is what I did in my PhD, and what the whole point of the slide was that by understanding the molecular biology and genetics, we might have better predictors of treatment. Now I think I could still use this slide in a talk that I could give today. And the other thing I talked about was that perhaps each tumour would be different for each patient and it require a different treatment regime. So depending on the genetic defects in each of the tumours, we might have a different way of diagnosing cancer. So this is very modern thinking. That was very modern thinking for the day, and is now the norm. And people like Ben Lawrence and Chris Print, of course, talk about this thing all the time. But it was interesting to see that the New Zealand Medical Journal didn't think that was worth a bar of salt in the end. So I became a lecturer in reproductive science, National Women's Hospital, surrounded by excellent people, including Mont Liggins, and my first head of department was Gillian Turner and John France, who were both great supporters and head of departments. We had Larry just down the hallway. Larry and I have been good friends for years. In order to recruit students, we decided we would just dress ourselves up a little bit. So this was just to show that what sort of students we wanted to attract to work at our laboratories. So Larry and I have worked together on a number of projects over the years and has been a great colleague and workmate. Over the years, had many excellent students. Debbie Prendergast worked in my lab for many years. We've got Anita here tonight, and Debbie, obviously, and a few dinosaurs. I'm not quite sure. And so many students over the years that have really added excitement and spice to the research life. Just want to talk about a few students in particular. So Chris Print has been also a wonderful colleague. Chris is actually a bit of a pain. Before he turned up, all the students thought I was wonderful. I always, I'd do things on time and I'd read their papers and whatever and get it back to them. But Chris, they said, why can't you get them back as fast as Chris does? Why can't you email me at 3 o'clock in the morning and get it done the next day? Chris is such a wonderful guy and so hardworking and much more intelligent than me. So you can hear from him next week. He can't make it tonight. I've already sent his apologies. We've also had another new arrival, which is Lindsay. Lindsay's really spiced up Larry and my life. She's been a great colleague, and she needs to be congratulated for teaching reproduction and taking the practical as well as the theory. So now, had many students over the years. So you might notice that most of them are female and very attractive. So here's Angel, and we're of course the Great Wall of China. So Angel and I had a great trip there. This is Roseanne. We're in Spain. What you might notice is that the students, some of them a little bit inappropriate, perhaps, and perhaps a little bit too cosy. But Mohan, it's never going to happen. So Mohan is one of the few Y chromosomes we've ever had. He's actually got a major problem. And I looked in the statutes the other day, and I don't know if you can read down the bottom here, but it says So these are the faculty results from the Round the Bays run. You can see that he's actually 2.5 minutes behind his supervisor, so it's unlikely he's going to be awarded his PhD, but maybe next year, Mohan. Now, I've just put up this slide I'm going to talk a little bit of science for about 10 minutes and then finish with a little bit about what other things I do in my life. My biggest problem is lack of focus. My world consists of genetics, cancer and reproduction. I just thought I would just note down the different areas that I'm involved in, and a lot of people in the room, and some people in the room I noticed before that aren't even on these lists, so it's a little bit embarrassing. But I've had, I guess many of us should learn to focus in life and perhaps just do one thing at a time. I've always dabbled in too many things. It's very hard to keep up to date with everything. But I've had many great projects and relationships with people in the faculty and, of course, many students over the years. I'm just going to go through a little bit of research and just highlight some of the things that I do in life. So this is the slide that I show every student that I teach. Prue's nodding ahead. That all human disease is a mixture of genetics and environmental factors. Some things are purely genetic, like Huntington's disease, inherited breast cancer. At the other end, some things are purely environmental, perhaps cigarette smoking and lung cancer is an example of that. However, even lung cancer and cigarette smoking, as Rob's up the back, that there is a genetic component of that as well. So underlying every human disease is my mantra. There's a little bit of genetics and environment, but of course it's the interaction of the two that brings about disease occurring. So this is a family that I studied many years ago, and what you can see is ovarian cancer occurs in nearly every generation of that family. So this is a pedigree, so the oldest people at the top, the youngest one at the bottom. Nearly every person in that family had ovarian cancer, often at a younger age than they should. And of course this is a perfect example of a cancer syndrome, and this family from memory was a BRCA1 mutation, which we found. I just want to talk about one project that came about. I'm supposed to be the Associate Dean of Research, and we're talking about sort of planned research and that sort of thing. Well, a clinician, Neil Johnson, came to see me one day. He knew that I worked on ovarian cancer. He said, I've had this funny family come and see me. The young woman came to the clinic with her mother, and with her periods had stopped at age 31. Her mother said, well, the same thing happened to me. My period stopped quite early, and something happened to my identical twin, non-identical twin sister over here. And also something strange happened to grandmother, about the same age, and your cousins were also unusual. It was the first time the woman had ever was aware of that problem running in the family. So when something runs in a family like that, to me, often might mean that there's a gene that's running through that family. So I put that to one side, and the very next day we got an email saying, anybody interested in a project on menopause, please write to the bread company, Bergen, because they were launching a new soy and linseed bread for menopausal women. And so for every 10 cents that you spent on the bread would go to a research project. And so the two things happen, an interesting family, the bread sponsorship appearing. So I applied for that project grant and got it for $10,000 and started off a whole new life of research. We went on to find a large number of genes that were present in these families. I won't go into these in any detail, but you can see some familiar names in here, including Cindy Farquhar, who's in the audience. So we found that this gene called inhibin was involved. We found another gene called FOXL2 was also involved in some of these families where we saw cancer running throughout the families. The other thing I did is that when I first made contact with a lot of the women that wanted to be part of the project, it was before the days of the internet, and they would tell me their life stories and want to know if they were a freak or the only person like that in New Zealand. And so as I collected these women for my research project, I realised there was an enormous need out there for good information. So I used to email them to them or post them and write to them, and we set up a little support group and actually had meetings together where the woman felt that meeting somebody else with the same condition actually changed their lives completely. Nowadays, of course, this has become a support group online. We don't meet together because we can do it virtually, and women are now accessing information that's really, really valuable. So for a non-clinician to be actually do something useful for women and their condition has been an enormous highlight of my research career. We also work on ovarian cancer, and a lot of my early research was on the more common epithelial cancers. But again, I got sidetracked to a project where a couple came to see me saying they had a rare type of ovarian cancer and there's very little research on that topic. Would I be interested, they were raising money by doing fun runs and things, would I be interested in looking at that particular rare tumour called a granulosa cell tumour? There are two forms of this, an adult type and a juvenile type. The adult form tends to be in older women and the juvenile tends to be in younger women, although there's a lot of overlap in the middle. So it's very dangerous to study a rare tumour in New Zealand because funding is always difficult. However, they were able to provide a few $1,000s every year, which made the research office, we have a Tracy Swifters, not very happy with me because these little grants would dribble in. What we were able to do is, because it's a rare tumour, we collected a number of historic tumour blocks, formalin-fixed, paraffin-embedded tissue. We had a little bit of tumour inside them, and we're able to look at certain different genes. This one we're looking at in this study here was called FOXL2, which you mightn't have noticed was involved in premature ovarian failure as well as ovarian cancer. So we were able to publish our findings on this just very recently, and we're now one of the leading labs in this rare type of cancer, although it's becoming increasingly difficult to fund. We made the observation that the single gene called FOXL2 caused both premature ovarian failure, which is the cells around the follicles not growing very well, and also a mutation in the same gene, also called a tumour, where the cells are growing out of control. We actually don't understand how this can happen. Same mutation causes infertility and lack of cells to grow, and an almost identical mutation causes the tumours to grow out of control. It's probably because the germline mutation, the mutation that you're born with, and is probably in the cells right from the very beginning, causes the abnormality of premature ovarian failure. Whereas this happens later on in life, it may be that the cells are already growing and growing out of control, although we don't know the answer to that. And fortunately, I've got a great student called Angel, again, who's looking at this, and she's also trying to find better prognostic differences between the adult form and the juvenile form. And she's looking at some very interesting molecules called microRNAs that seem to be different between the adult and the juvenile. She's also got a funny thing for dragons and likes to pet them as well. I'm also well known for all students for talking about infertility. So my job in first year is to scare all the female students into having children early. What I talk about is that there are a number of women that only have a limited number of follicles from life when they're born right through to menopause. The decline in fertility in the 20s, most students will know the magic age is 24, is the best age to have a baby. After the age of 30, we see a decline and then a more rapid decline. And most women can't guarantee to have children over the age of 40. I had a medical student a few years ago called Nathaniel who came to ask me about a project, and I just read a publication. So why don't you copy what these people have done and study some of our own students and see what university students understand about fertility. So he had a 20-question questionnaire. He surveyed 683 students around the university, something we're not allowed to do anymore, I'm not quite sure why, about estimate the chance of a woman becoming pregnant if she has intercourse with a fertile male. The second question was estimate the likelihood of a woman using technology to have a baby. And what he showed was that this is what the literature will show, that as a woman gets older, the likelihood of having a baby declines. And all of our students overestimated by about 10 or 20% at every stage in life about how fertile they might be. And if they use technology, what you can see is that even with technology, as women get older, the technology doesn't work, and our students completely overestimate how fertile they'll be, even using technology. So I think the main outcomes of that study is that most students can identify that fertility declines in women, but students completely overestimate their fertility rates, and most of them overestimate the use of assisted reproductive technologies. The medical students were the worst for the use of technology, and the nurses also were similarly poor at being able to work this out. The only students that seem to do okay with this are the ones that have been lectured by me. So, okay. So we also have another very exciting project involved with Leslie and John Thompson, Mohan, and this also involves Claire Wall and Ed Mitchell. We're looking at a study that Leslie's been involved with, where we've got 1,200. This is, I tried to summarise Mohan's thesis in one slide, so I've taken a few liberties. What we had is 1,200 6-year-old children. We had information about the mum, and all through her pregnancy, got information about the dad. We were able to extract DNA from the children and have their DNA available. We also had understanding of their diet and their exercise and sleep. And what we're trying to do is work out which of these contributed most to the obesity of 6-year-old children. So Mohan's swimming in a sea of data at the moment. One of the interesting things we did is just compared BMI, which is a common indice of obesity, and also we are able to measure the percentage body fat. We see that's not a perfect relationship between the two, but in general, it's if you have a high body fat, you also have a high BMI. When he looked at the genetics of both of these processes, he realised that a number of genes were significantly associated with both percentage body fat and BMI, but not much overlap between the two. So probably we're looking at, when we're looking at obesity, we're looking at a range of different genetic markers that may be influencing the outcome of these, and only a little bit of overlap in the middle. Mohan's got a few years work left to come up with a real solution to obesity in New Zealand. Another interesting snippet I just want to talk about is in breast cancer, one of the most significant successes over the last 20-30 years, if a woman has estrogen receptor staining in breast tissue, then she's likely to receive chemotherapy. If she has 1% or more, she's likely to receive slightly more modern endocrine therapies like tamoxifen or aromatase inhibitors. So the cutoff is made between 0 and 1%. So if you've got no staining, you get chemotherapy. If you get 1% or more, then you can have endocrine therapy. But we know that the staining is not bimodal, and that it's there's intensity is linear. And what Anita in my lab, who's been one of the longest stayers and deserves a medal for perseverance, and of course the brain power behind it is Chris. What we realised that women that are ER positive here, and we measure this is measuring the amount of estrogen receptor that's present in each of their tumours. This is in close to 1,000 tumours. There's a linear representation along here. So some women will have very low levels of estrogen receptor, even though they're diagnosed as ER positive. And even so, some women that are called ER negative, some of them will have high levels of estrogen receptor expression in their tumours. And we know that about 30% of women don't respond to endocrine therapies, and some women become resistant. So we sometimes wonder from some of this work whether or not we can make a better diagnosis on a mixture of pathology and more modern molecular genetics. So that's just a little spice of what we do in the research lab. I just want to talk about one other piece of research in the last 5-10 minutes that I've been involved with. This is Peter Shepherd, or if you're Minister Joyce Shep, who runs Queenstown Research Week. And one thing that didn't go into my promotion application is my contribution to a field of research called fashionomics. So this is a competition down in Queenstown whereby researchers get to dress up in clothing from the laboratory. And what happened a few years ago is that they decided they needed another night for yet another dinner for all the important speakers. We'll cancel the fashion competition. And I said, that's not fair. The students just love it. So Peter said, well, I'm in charge if that's the case. So I thought I couldn't be in charge of an event if I actually didn't compete in it as well. So the whole point of fashionomics is that you've got to scan the laboratory, find some equipment, and produce a garment, find somebody brave or presentable to actually model the above item. Alternatively, just tell your supervisor that you'll quit unless they don't do it. And then you come up with a commentary. So over the years, I've embarrassed myself in many, many different ways. And this is also, John, you probably shouldn't see, this is a lot of faculty resources and time going into preparing some of these costumes. This is one where I was a mutant sperm. This is a mitochondrial belt here and acrosome cap. This was the egg. Annette Lasham decided not to come tonight, thinking that they might. This one is almost too embarrassing to talk about. I was a uterus, and this is sperm that stuck to the uterus. I won't talk about the rest of the outfit. This is another year when it was a little bit more dull, but this is actually Carrie Mullis, who Nobel Prize winner, who invented PCR, a very important lab technique. He was so taken by what we were doing, and he wanted to take it back to the States and get everybody to do it. This is probably my best outfit. So Sheree Lenkeron is the main architect of this one. This is filter paper from the lab that's been dyed with all the different stains that we use in the laboratory. She spent weeks doing this, probably when she should have been writing papers and writing grants, but it won the competition and she got a free trip to Fiji as a result of it. This was last year when I was a genetics conference and a reproductive conference and a plant conference. So I was Andrew the bisexual plant, and I had Angel the bee right behind me. But probably my best effort, and I won't go, I see I'm running out of time. This is talking about the differences in the old days when we had to do everything for ourselves. We had to prepare competent cells, pour our own gels, throw out the radioactivity, and we'd have to photocopy things from the laboratory if we were lucky. Then I became Andrea the skanky PhD student. Where they don't turn up into the lab until quite late. They check everything on Facebook, Twitter, and online TV all day long. Nobody seems to mind. It's been a long week. Will be at least 3 days of work, sending off all those samples to be assayed. She's waiting for the bioinformatics guy to analyse the data. She even had to go into the lab the other day. It was really inconvenient. She couldn't check her Facebook. So anyway, Googling, she had some problems. Googling it to give her an answer. The only solution was to buy more expensive kits. So all the old people that heard that understood it more than the young ones. So I'm just going to finish, hopefully, in the next 5 minutes, hopefully. So teaching is an important component of what we do. This is the North Korean election, where you convince the students that you're wonderful, and hopefully they'll give you all scores on that side, and so that you're really great. Most of the students will recognize my slides. So this is where I use Comic Sans and this sort of yellow colouring. Thanks to cyberbullying, I've now changed my style, and it's now Arial. I will show that. So one of the most amusing things that happened a few years ago is the students will write nice things about you, and so they often say he's friendly, he's a good lecturer, and overall his lecturing is very good. But then on the next page there was but the only mistake that the student made was that I was marking their exams at the same time. And their writing was rather distinctive, so I knew who the student was. A very attractive woman. But one of my students, I thought, well, maybe I'm not so old after all. But one of my students pointed out that she started university as a he. So I just want to finish in the last few minutes, because most of us have had a very long day, just talking to you a little bit about what I do in my spare time. I still like to get out in the countryside and do lots of active things. This was a few years ago doing the Coast to Coast, which is a little jog over the South Island. Still do a lot of running around the place and go on various events. It's always more fun when you do it with your family, and it's a great way of brainwashing them when they're young that this is a normal thing to do. We live out in the countryside a little bit, about 50 minutes away. It's a way that I get away from work and try and unwind and relax just a little bit. Been a great place to bring up kids. Here's my son Levi with a little lamb. We've had lambs born just in the last week, but great for them to learn about life and death. And some of them win competitions, so the kids have learnt which ones are winners. Ones that are losers, of course. Madeline's done a lot of breeding of animals around the place. Levi's had a lot of pets over the years. This is a turkey called Turcules. He's never been short of a girlfriend. And we do a few other nefarious things out in the countryside that might be, this is not Cecil. And some of you may realise I escape every few months and disappear into the mountains somewhere in the South Island just to try and get a little bit of peace and quiet and enjoyment of the countryside. So that's just a little bit about me. Thank you for being here and hearing a little bit of a story about my life journey. And I'll now pass you over to Phillip.
I'm Phillip Aspinall, the acting head of School of Medicine, and I'm actually standing in for Leslie McCowan tonight, who unfortunately isn't well, but she's sent loads of warm wishes your way and actually given me a few words to say on her behalf. I will be confirming to her that we've just heard a masterful inaugural lecture and that there were plenty of people present. It's really lovely to see the family, and thank you for allowing Andrew to enjoy your lives as well as his. Thank you. Some of you would have been coming along to support Andrew. Some would have been really piqued by the title, as I was, about the anatomy of the genetics of a professor. So I think we've all been entertained by a very deeply personal and professional journey, and I, found out a lot about Andrew, including your sense of humour and, but always your honesty. And I think you shouldn't apologise for all those collaborators. I think most of us in the faculty just frankly enjoy working with you and value your wisdom and really enjoy that relationship. So keep on doing just what you're doing. So you can see what a gifted teacher, supervisor, researcher, academic leader Andrew is, and what a gem he is in our faculty. So it really just remains for me to say, tonight's been a good example of a good genes interacting with an excellent physical and family environment. So good testament to that. So please welcome to the professoriate, and all of you can join me in thanking Andrew for all his work, but also his friendship and also for his wonderful lecture tonight. Thank you.
Thank you very much, everybody, for coming along, especially to family and friends who braved a rather cold night. But Andrew, that was a wonderfully entertaining lecture, and suitably for our inaugural lecture, our third inaugural lecture. It's only left to me to formally close the proceedings for tonight and remind everybody that on Thursday night we have Professor Peter Adams, who will be presenting his inaugural lecture in this lecture theatre. So thank you all for coming, and thank you again, Andrew, for a wonderful lecture. Thank you.